So welcome students to part 2 of chapter 9, and in this portion we'll be talking about the neuromuscular junction, which is the junction between the motor nerve and the skeletal muscle, and then how the action potential, or that order, the electricity coming from the motor nerve, how does it reach the muscle, and how does the muscle actually contract. So first off, we want to talk about the structure or the anatomy of the neuromuscular junction, which is basically the junction between the nerve and the muscle. It is also known as a myoneural junction. So again, either way we're talking about the same structure. This is a kind of synapse. So a synapse is where a nerve connects to something else. That something else could be another nerve, it could be a muscle, could be a gland. In this case of the neuromuscular junction, it is the junction between a motor nerve and a muscle. And notice I keep on saying motor nerve because it's obviously not a sensory nerve. A motor nerve is the nerve that brings in, it is the efferent or the efferent neuron that brings the order to the muscle to contract. And where they meet, again, is known as your neuromuscular junction, and is made out of these different parts. So you have the end of the motor neuron, and that is known as the terminal knob, K-N-O-B, or known as the axon terminal. And it is also made out of the sarcolemma of the skeletal muscle, and you could see kind of like these waves. That is known as your motor end plate. So where on the sarcolemma of the skeletal muscle, where it meets these axon terminals, that is known as your motor end plate. And you can see a bigger picture over here. So this is the terminal knob or the axon terminal. You can see some mitochondria in there, and you can also see these vesicles. These vesicles have a very important chemical known as acetylcholine. And acetylcholine is the neurotransmitter of the neuromuscular junction. So all of our skeletal muscles, in order for them to contract, they are these nerve endings are going to release acetylcholine. And then there's a gap between the nerve ending and the muscle, and that gap is known as the synaptic cleft. The synaptic cleft. On the muscular end, so again, we have these waves, and on them you will find receptors. These are acetylcholine receptors, also known as cholinergic receptors. So your parts, again, you have your motor neuron, the motor end plate, and that again is the part of the sarcolemma. There's the synaptic cleft, and that is the gap between the motor nerve and the sarcolemma. There's the synaptic vesicles, and these are the vesicles inside of the motor end, or the motor neuron, that are full of acetylcholine. And then we have our neurotransmitter, which is acetylcholine in the case of the neuromuscular junction. So let's see what happens. In order for the muscle to contract, again, the order comes on that motor neuron, and somehow it has to cross over from the motor neuron to the skeletal muscle. And this is how things happen. This right here, again, is the end of the motor neuron. You get the order or the action potential that reaches the synaptic terminal, or reaches that axon terminal. And when that happens, calcium channels here open up. Calcium will rush into the inside of the nerve, and that would push these little vesicles to release the acetylcholine into the synaptic cleft through a process of exocytosis. So again, motor nerve, the motor order comes to the end of the motor nerve that opens up calcium channels right here. Calcium rushes to the inside of the neuron, so there's an increase in the intracytoplasmic calcium levels. That leads to the opening up these vesicles and releasing of the acetylcholine into the synaptic cleft. Now, acetylcholine pretty much swims across that synaptic cleft and attaches to the receptors that are on the sarcolemma. These receptors, when we activate those cholinergic receptors, that is going to open up sodium and potassium channels. And now we have, that electricity that used to be, that was coming from the nerve has now been transmitted to the muscle side, to the muscle end. And that is due to, thanks to the acetylcholine activating those receptors. Now, now that the electricity is on the muscular end, that is going to open up. Remember, we said that calcium is stored in the sarcoplasmic reticulum. Now, that when the electricity reaches the muscle end, that will open up the cisterns of sarcoplasmic reticulum, releasing calcium. Now, this is happening inside of the muscle. So now there is an increase in intracytoplasmic calcium inside of the skeletal muscles. So we'll see how that is going to lead to your contraction. I guess in the next couple of slides. Okay, so little clinical application on this whole process. So we've seen that in order for a muscle to contract, you have acetylcholine that has to attach to acetylcholine receptor. Some individuals have an autoimmune disorder, which technically means that the immune system is unable to recognize its own cells, the cells of the body, and starts making antibodies against that attack that person's own cells. That's what an autoimmune disorder is. And in this case, in myasthenia gravis or MG, the person is making antibodies against those cholinergic receptors, against the acetylcholine receptors that are found on skeletal muscles. So you could imagine that there is now, acetylcholine comes out, but there are no receptors to attach to. Okay, so the autoimmune disease will lead to the destruction, the deactivation, and the destruction of a lot of these receptors. So sometimes, there are some receptors that kind of escape the attack. So maybe about 1/3 would have normal, or they would have about 1/3 of normal number of acetylcholine receptors. But these individuals will definitely have muscle weakness and muscle fatigue because of the loss and the destruction of the majority of their cholinergic receptors. So how do we treat the disorder? In general, autoimmune disorders are treated by several different things. You could give them immunosuppressant drugs, so you are trying to suppress their immune system, but you could see how that could translate into, more infections, and they become prone and are at high risk of other infections. Or you could give them antibodies to destroy those harmful antibodies. So if their antibodies, their bodies and their immune systems are producing antibodies, well, why don't we give them synthetic antibodies to destroy those harmful ones? Or we can do a plasma exchange. A plasma exchange is basically so remember that antibodies are circulating in plasma. So in a plasma exchange, you would technically, drain the plasma of the patient, filter out the antibodies, and give them the plasma back, now fresh, minus the antibodies. So this is obviously done in a very gradual way. It's done over hours. We don't literally drain all of the plasma out of the body at once. Okay, it's a very gradual procedure. And our last option in this case would be to give out a drug that inhibits acetylcholine esterase. So hopefully you realize that an ACE means that this is an enzyme. The whole thought behind this is that acetylcholine breaks down by an enzyme known as acetylcholine esterase enzyme. So we will give them a drug that inhibits that enzyme. So we are upping up or keeping the level of acetylcholine and preventing its destruction, and trying to eliminate or trying to kind of compensate for the low levels of receptors. Now, moving on to the rest of the story of muscle contraction, we have what is known as the excitation-contraction coupling, which is basically the coupling between the excitation of the muscle and the contraction of the muscle. So how is it that now we have excited or stimulated the muscle fiber? Now, what happens in order for the muscle to contract? So we already talked about, the increase of calcium levels because they were released from where they were stored in the cisterns of the sarcoplasmic reticulum. We already talked about in our previous lecture in part 1 how troponin and the tropomyosin complex cover the binding sites on actin. All of that is found during relaxation. So what happens when the muscle is stimulated? Well, calcium is released. Okay, it increases in the cytosol. So there's an increase in the cytoplasmic calcium levels. Calcium will bind to troponin. And troponin, remember, that was that protein that kind of looks like a rocket. It'll change its shape. It changes its configuration. And now troponin and tropomyosin get out of the way, and that exposes the binding sites on actin. Now the myosin head is able to attach to actin, forming what we call cross bridges. So the attachment of the myosin head to actin is known as a cross bridge. You can see this whole scenario kind of like in this, cycle, this cross bridging cycle, and we will start all the way up here. So this is at rest. So at rest, you have your myosin head. There are the troponin and tropomyosin, and those are covering, again, the binding sites on actin. When calcium comes in, okay, so calcium is released from the cytoplasmic reticulum. Calcium will attach to these troponins, these light blue proteins, and they will change their shape. And now you can see those little purple binding sites. Those are the binding sites that myosin head will attach to. So you can see now the myosin head attaches to these binding sites, and that is known as your cross bridging. Now the myosin head is going to flex, bend, pulling on the actin protein like we've seen in that previous video. In order for, and that leads again to the sliding of actin on top of myosin. In order for that myosin head to let go, we need ATP. So myosin head will stay attached to the actin binding site unless it is given an ATP molecule. When an ATP molecule is handed over to the myosin head retracts, and it stores all of that energy. It'll break down the ATP into ADP and phosphate and store that energy in the myosin head. And that is known as cocking of the myosin head. And it is related to the term of cocking the myosin head is related to like an erection of the myosin head, where the myosin head is now ready, has a lot of energy, and will go ahead and attach to another binding site if the binding sites are exposed. Okay. So that is what happens, and this is like the whole cycle of how this occurs. This again is the rest of the cycle. I just have it on there twice, just to show that what happens in order for a muscle to relax. In order for the muscle to relax, well, we have to get rid of calcium. Okay, so the whole scenario really depends on the presence or absence of calcium. So in muscle relaxation, we want to get rid of calcium, and we do that through a pump. It is an active transport of calcium. It's kind of like a vacuum that sucks calcium back into the sarcoplasmic reticulum. When we do that, these troponins, blue things, go back into the position. So do tropomyosin. Now these binding sites are covered, and that myosin head has nowhere to attach, and it just stays there. And the muscle relaxes. So the model that we talked about is the sliding filament model, where we talked about the sliding of the actin on top of myosin. And that's what this model or theory talks about. And when that sliding occurs, the sarcomere will shorten. So remember, we said a sarcomere is from one Z line to another Z line. When the filaments slide on top of each other, you can see that these zones gradually decrease in size or decrease in length. So those will include your H zones and your I bands. The sarcomere even shortens. okay, up until where pretty much until you see that there is no more space. So there is a limit to muscle contraction. Muscles can contract, but as soon as there's no more shortening to occur, then you've reached that limit for that muscle. So the overlapping can only reach a limit. They cannot, further overlap on top of each other. What happens in relaxation? We kind of touched upon that a little bit, that the calcium pump or that vacuum is going to pump calcium back into the sarcoplasmic reticulum. But what makes that happen? It really goes back to acetylcholine. So as long as acetylcholine, ACH stands for acetylcholine, as long as acetylcholine is activating or is stimulating the receptors, then contraction keeps on going. So we really have to get rid of acetylcholine. There's different ways of doing that. So acetylcholine can pretty much just diffuse away from the receptors. But there's an enzyme as well, and we already talked about that, the acetylcholine esterase enzyme, and we mentioned that in certain diseases like myasthenia gravis, we can give drugs that would inhibit the activity of that enzyme, to increase the amount of acetylcholine found in the synaptic cleft. So when acetylcholine esterase breaks down acetylcholine into its two components, acetyl and choline, the muscle impulse will stop. Okay, and at that point, calcium will be pumped into the sarcoplasmic reticulum. Your troponin-tropomyosin complex reverses back into its position, covering the binding sites on actin. And now myosin head has nowhere to attach, and that leads to the relaxation of the muscle fibers. So what are the energy sources for contraction? Obviously, it is ATP. So we do have a small reserve of ATP. We also have creatine phosphate, and that is how we store phosphate. So remember that in order to make ATP, we are going to bind ADP to a third phosphate. So ADP is adenosine di or 2 phosphates, and add to it a third one, okay, to make your ATP. Phosphate is stored as creatine phosphate. So phosphate, or that PO4 molecule, is a very small molecule. It could kind of wander around and get lost. So that we do not lose it, we bind it to creatine. Okay, so creatine phosphate is how we store phosphate. And you can see how breakdown of what happens when ATP levels are high. We are going to go ahead and break down that ATP into ADP and phosphate, and that will release the energy that was stored in that energy bond. And again, so that we do not lose the phosphate, it is attached to creatine. Okay, so creatine phosphate, again, is your phosphate storage molecule. Now, when cellular ATP is low, we want to build that ATP, those ATP levels back again. We will just bind the ATP back with the phosphate from creatine phosphate to make ATP. And creatine is just, recycled. We can use it all over again. So this is what happens, say, when we're exercising and we need energy. Okay, we're breaking down ATP, releasing the energy and using it for exercising. In between, so when we're relaxed, the muscle needs to build up all that ATP that it used, replenish its ATP supply, and that's how it does it. So what different kinds of cellular respirations do we have? So cellular respiration is basically taking a glucose molecule, breaking it down, and releasing, using it to make ATP. There are two different kinds of cellular respiration. One is anaerobic, that does not need oxygen. And that is known as glycolysis. So glycolysis, okay, means to break down glucose. That will happen in the cytoplasm, and it produces a very small amount of ATP. It's pretty much producing 2 molecules of ATP. So your anaerobic phase will make ATP, but is definitely not enough to make the amount of ATP that a muscle would need to contract. So when we go through this anaerobic phase and we need more, we could go ahead towards this aerobic phase. And this is when we need oxygen, hence the name aerobic. It happens in the mitochondria. So remember, the mitochondria was your powerhouse of the cell, and this is where most of the majority of ATP molecules are made. The aerobic phase is made out of two different stages. There is the citric acid cycle, also known as Krebs cycle, and there's the electron transport chain. Both of these happen inside of the mitochondria. Because these are aerobic phases, they need oxygen to make sure that the skeletal muscles always have a good oxygen supply. Oxygen is stored in the muscle in the form of myoglobin. Myoglobin is the sister of hemoglobin. So globin is a protein that is able to store oxygen. If it's in the blood, we will name it hemoglobin. If it's in the muscle, we'll name it myoglobin. So they are related to each other. It's just where they are in the body. And again, myoglobin is how you store extra oxygen so that the muscle will be able to go through this aerobic phase and produce a lot of ATP. Now, what is the oxygen debt? So let's say, for example, somebody like me that unfortunately doesn't exercise enough. Like, I wake up in the morning and I say, you know what, today I am going to run 2 miles, okay? Or I'm going to exercise for a whole hour. Come tomorrow morning when I wake up, I'm going to be very sore. That soreness is because my muscles are not trained enough. They do not have enough mitochondria to produce the amount of energy that I needed in order to exercise for that whole hour. I am not trained, I'm not prepared, and my muscles are not prepared. So, going back here, okay, in a trained individual, their muscles have enough mitochondria where they would go through anaerobic phase and then aerobic phase. In an untrained muscle, like myself, I would go through anaerobic phase, but because my muscles do not have enough mitochondria, because again, either I didn't warm up, I am definitely not physically active or physically prepared for that amount of activity. So the number of mitochondria in my muscle cells weren't enough to keep up with the demand of that whole 1 hour of exercise. So this glucose will break down, and instead of going to the aerobic phase, it ferments, it goes bad and produces what is known as lactic acid. And it is that accumulation of this lactic acid that led to the soreness that I would feel the next morning. Now, lactic acid obviously is a toxin, or is, it's not poisonous, but it is something that the body wants to get rid of. Okay, it is causing soreness, it's painful, irritating the nerves and causing that pain. So that lactic acid is going to be transported through the bloodstream to the liver. Now it is up to the liver to change the lactic acid back into glucose. In order to do so, the liver needs a lot of oxygen, and that oxygen needed is known as the oxygen debt. Okay, so the oxygen debt is the amount of oxygen needed by the liver cells to change that lactic acid that accumulated, okay, due to the fermentation, to change it back into glucose. It also want to restore the ATP that I've used for that amount of exercise. So remember, I broke down a lot of ATP, and I need to restore that back. So I need to restore my muscle ATP and the creatine phosphate, which is how I store phosphate. And all of these processes need oxygen. So again, it's kind of like the liver cells are borrowing extra oxygen from the blood in order to take care of all of these problems. And that again is known as the oxygen debt. Okay, now what is the anaerobic threshold? So what is the threshold of when this would actually occur? When would the metabolism shift from aerobic to anaerobic? Again, That would be during strenuous muscle exercise when a muscle is not prepared for the amount of exercise done. That is why if you exercise, you always want to warm up first to make sure that the muscles are getting enough oxygen, enough blood supply. So when the above systems cannot supply the necessary oxygen, okay, again, the pyruvic acid, instead of going through the aerobic phase, it'll start to break down anaerobically and ferment into lactic acid. Here you can see kind of like, how this cycle works. So remember, glycogen is how we store glucose in our skeletal muscles and liver. But this, this whole story at first starts in the skeletal muscles. So the skeletal muscle is exercising. It'll break down glycogen into glucose. Now that glucose will go through the anaerobic phase first, maybe a little bit through the aerobic, but at some point where now I am all out of mitochondria, at a point, the pyruvic acid, instead of going down to Krebs cycle, it will ferment into lactic acid. Lactic acid will be removed from the muscles by the bloodstream and will go to the liver. The liver then will change that lactic acid back into glucose. Okay, and again, that needs a lot of oxygen. That oxygen that the liver borrowed is known as the oxygen debt. So what is muscle fatigue? Muscle fatigue is the inability to contract muscles. So you've seen these marathon runners where they're almost at the finish line and they're very tired and they're just at a point where they can no longer contract their muscles. So these are common causes of why that might happen. Either there's a decrease in blood flow or there's an electronic imbalance. We obviously need calcium to contract our muscles. We also need sodium and potassium. So any imbalances in these electrolytes can lead to muscle fatigue. Or there might be a loss of desire to continue exercise. Maybe it's just that, the stress of the process. And that's why when people start cheering them on, that kind of energizes them to continue. There might be the accumulation of lactic acid, but that is a little bit controversial if it actually has a role or not. But these are all, different hypotheses and different reasons why muscle fatigue would occur. Muscle fatigue is very different than muscle cramping. So we've all had a muscle cramp, which is an involuntary skeletal muscle contraction. And that occurs due to a change in the electrolyte concentration surrounding the muscle. So in the extracellular fluid that is surrounding the muscle. What about heat production? So when we are breaking down glucose, producing ATP, well, there's a lot more energy being produced, okay? Burning glucose produces tons of energy. Most of that energy actually is going to be released in the form of body heat. So muscle cells are a major source of body heat. The muscle cell that produces most of the body heat is actually the cardiac muscles. And that makes sense because the blood is right there inside of the heart. And then the cardiac muscles are contracting all of the time, producing a lot of body heat, producing thermal energy. And that energy will be distributed through the bloodstream to the rest of the body. Now, the majority, again, of the energy produced due to muscle contraction is released in the form of thermal energy. About less than half of that energy is going to be used to make ATP. Okay, like I said, the blood will transport the heat throughout the rest of the body. Now, at this point, I'm going to go ahead and stop this portion, and I will see you folks in part 3.